Okay, I think we'll get started today on the Proteoform Thursday. This is a monthly seminar featuring the latest research from leading scientists in the top-down proteomics and proteoform biology. It's presented by Consortium for Top-Down Proteomics. Top-Down Consortium has a special mission to promote the young scientists. So this Proteoform Thursday also used to promote many young scientists in early career stage and help them succeed in the future. So today's speaker is David Roberts from Stanford University. So before introducing David, I want to go to a couple of business slides. So first of all, I'd like to thank our annual sponsors, the consortium major sponsors, Thermo Fisher, Bruker, SciEx, and Eli Lilly, as well as the consortium supporters, Agilent, Pfizer, New Objective, Immunogen, and MSC. So if any of you in the audience are associated with a company, we would appreciate if you let your employer know this great opportunity to support young scientists and become a sponsor of the consortium. Okay, so I also want to take this opportunity to remind you that we have our third International Top-Down Proteomics Symposium scheduled for 2025 in August 26 to 28 at the University of Hamburg, Germany. So if you are interested in top-down proteomics, also like to visit Germany, it's a great opportunity. And the registration and abstract submission are not open. So this will have, both the emerging technologies integrating data types, drivers, biology, and clinical research and application to life science. It also included two short courses. Also, I know many of you will be going to ASMS soon, and everyone should be busy preparing your either oral or poster for ASMS. So I want to also draw your attention of the Monday evening workshop on top-down proteomics. This time, they're going to focus on the software and data analysis strategies for getting the most out of your data. I'd like to thank Caroline Lowe for organizing this, as well as Corinne Lotomarsky. And as always, we have the short course on top-down proteomics. Oops. Okay. Saturday, May 30th, as the top-down proteomics, mainly just the lectures and introduction, and the second day, June 1st, on the data analysis and the visualization. If you are still interested and have not registered for the short course, it's last time you could continue to register for it. Also on Wednesday, we have the oral section on top-down proteomics and posters. Monday and Tuesday, anti-proteins, anti-protein analysis. So far, we have almost more than 90 presentations on the top-down proteomics, and I hope this number will keep growing. I also want to say that the next seminar speaker is Juan Vizcano from European Bioinformatics Institute and EMBL. So now it's the time to introduce David. So I have a great pleasure working with David for quite a while in his PhD, and David is very uniquely trained young scientist. So David grew up in Bahamas, and it's a very beautiful place. He came to United States to study first in the University of California, San Diego, and he earned a BS there. Then he chose to come to UW-Madison to pursue his PhD. And so he did a joint PhD between my lab and Professor Song Jing's group at UW-Madison. So In Jing group, he learned to become a material chemistry, nanotechnology expert. In my group, simultaneously, he becomes expert in mass spectrometry, particularly in native mass spectrometry and glycol biology. So impressively, David actually was offered a faculty position before even he graduated, but he decided to do a postdoc under the Nobel laureate Carolyn Bertozzi to strengthen his chemical biology training. So he's now in the Department of Chemistry and at the Stanford University. So his title today is Guiding the Rational Design of Protein Assembly by Native Mass Spectrometry. So here you go, David.
Thank you, Ying. So let's see if I can share this. Okay. Can everybody see the slide?
Yes.
Okay, great. Yeah. So thank you so much, Ying, for the very kind introduction. So as Ying mentioned, so today I'm going to be talking a little bit about some of the emergent science that you can imagine when you come from a mass spectrometry background and start moving into more biologically driven questions. And so I thought the title for today is a bit provocative, Guiding the Rational Design of Protein Assemblies by Native Mass Spectrometry. And the hope here is that I want to paint a little bit of a different view of how mass spectrometry techniques can be leveraged, not necessarily for traditional purposes, but also to kind of enable the biophysical characterization of a lot of complex biology. And so being a postdoc here in Carolyn Bertozzi's lab has really opened my eyes to a lot of the kind of emergent things that you could imagine in biomedical science and translational science. And so I feel that there's a lot of ripe mass spectrometry to be played in a lot of these fields. And so seeing these connections and trying to build new bridges into other fields that otherwise don't have these kinds of insights, I think is an important part of how we can expand mass spec to the broader public sphere. And so starting off on this kind of conversation, I think we want to impress on everyone that no protein exists alone. And so the idea here is that when you think of protein biology or you think of the cell in general, you might imagine that you see various kinds of proteins all on their own, but in reality, most proteins are never actually isolated. They either exist in a tendency to be in some kind of connection, either an interaction in a true sense, or even just a self-interaction that we call an oligomer. And so what I'm separating out here on the left and right are the two major classes of protein interactions that we come to know. Homo-oligomers, which is interaction with itself, and hetero-oligomers, which is an interaction between two proteins that are different. And this word obligate refers to whether or not that interaction occurs stably in vivo, or is it a transient kind of state, non-obligate, and it serves a function. For example, some proteins that are extraordinarily essential, like this DNA binding protein you're seeing over here, AA representing two of the self-similar proteins coming together. This is an essential homo-oligomer, and they tend to stay well preserved. And in the case right next to it, this sperm lysin is essential for fertilization, but hopefully this isn't happening at all times. And so this only forms an oligomer when it's required. And so you can imagine that in biological context, functionally, when proteins are moving and talking to each other, this is kind of how the aspect of proteins actually carry themselves out. In heteroligomer cases, we actually have even more complexity because now it's not even just the same protein talking to itself, but another protein talking to something else. And so I'm writing this as AB to denote two unique protomers coming together either in an obligate case or a non-obligate case. And so you can imagine in the wider sphere, homo-oligomers tend to be a little bit better studied. And the reason for that is very clear. It's because these are exactly the same things, but just now oriented or translated or stacked in a kind of symmetrically preserved way. Whereas in the other case, in the hetero-oligomer sphere, protein A, protein B, who knows what they might be, but also they tend to be rather complicated because we don't have really good roadmaps to understand at the post-translational level how things are going to be arranged. And so this is actually probing further into a deeper question, which is the idea of how we can think about not only looking at the native biology of how things come together, but also understanding how we might be able to leverage this concept of protein self-assembly and thinking about it from the concept of a minimal scaffolding. I'll get to that and show you what I mean by this. But you could think about it from a chemist's perspective. When we're building these things from the ground up, we really have kind of two major structure types that exist naturally. This little helical thing you're seeing over here on the left is a classic alpha helical notation. And this little sheet you're seeing here that's kind of oriented in one way, that's what we normally call a beta sheet. Now, these two things exist on their own. You form multiple different various kinds of proteins, as you can see here, ranging from collagens, lysosomes, and so on. There's ways for these things to come together in a functional and productive way. And there's also ways for these things to come together in an aberrant way. You can imagine when you're scaling a lot of beta sheets together, what you're actually mostly doing in nature are making amyloids. But if you think about it from the point of view of fibrils and other things, these actually make up an essential part of your extracellular matrix. And so they have huge, reasonable ways of being coming together and having some function. And the more and more we can study these underlying molecular mechanisms, the more this gives rise for us to kind of understand the basic principles behind how biomolecules come together, how they fold, how they associate, and how we can better leverage them for new applications. And so the idea is sequence to structure, to function, to process, to biomaterial, and then to application. And so the better we can kind of put a molecular microscope to each of these individual cases, the better we'll be able to understand native biology, but also leverage this for new purposes. And directing back to the concept of heteroligomers, there's actually so far only two known major kinds of heterooligomeric motifs, and they're both alpha helical. The first one I'm going to show you up here on the top left is what we call a coiled coil motif. So this is essentially two alpha helices coming together and how their side chain pairings are occurring. So if you stare down the bottom of this duplex you're seeing over here, you can imagine like the bottom of the barrel, and you try to annotate some of the side chain residues in a kind of stereo projected way, it kind of looks a little bit like this, where at certain repeating units, there's side chain pairings that occur. And so these stereograph projections, these kind of circular helix diagrams, are kind of a very well understood way of rationalizing how these two things come together. So this is a protein A, this is a protein B, but they're technically different. They interact at the side chains. The other kind of natural example that we can find in biology are these collagen-like motifs. So these kind of make up some of the proteins you tend to see in your skin, and they tend to be very large, very complicated, they're heavily modified, but the underlying principle of their secondary structure are essentially either a group of homo alpha helices, which are same proteins coming together, kind of like this, or hetero alpha helices. And so I'm showing you here, this is a collagen heterotrimer. It's a bit of a bundle, but in reality, if you look down the center of where this extends, it looks quite similar to a coil-coil motif with just a slight modification. And throughout the last few decades, people who have been interested in trying to build new kind of designer motifs try to leverage what I'm calling here as a semi-empirical method. The idea is that you take examples from nature and you take examples from databases, so protein structures that we've well reported and accurately annotated, and we try to leverage this to design new scaffolds. But so far, all of the scaffolds we've been able to really come up with still rely on the basic principle of either coming in with an alpha helical coil-coil type motif or an alpha helical collagen-like motif. And so you can imagine that we're kind of limited in some way to really only taking what we've been able to easily extract from our natural environment. Now, very topically, the most recent Nobel Prize that's been awarded in chemistry actually went to Professor David Baker at UW-Seattle, and this went for protein design. And so it turns out, even if you look at modern de novo design approaches for creating heteroligomers, so this is a really nice little Rosetta structure here shown. So A, B, C, three different things coming together in a heteroligomer way. But if you look at the underlying motif, these themselves are still coil-coil for the most part. And the way that these are even designed in the Baker Lab and others is still kind of starting from some sort of template, which kind of relies on existing structure types. So if you want to imagine diversifying some residue, especially if you're wanting to do this in a very small or minor way, well, the kinds of structures that we can imagine or predict from those basic principles actually are quite limited. So although we have good resolution on how coil-coil or collagen-like alpha helices might behave, we actually have very low resolution and understanding of how small changes to protein side chains or modifications, or just one addition of a single post-translational modification somewhere, how is that going to affect the overall macrostructure? Turns out we don't have really good data sets for this. Importantly, we can't really predict too easily new kinds of designs in a de novo way when we have no real empirical data for this. So the point I want to stress here is that we have really wonderful, powerful computational methods that are even more so now racing to try to enable new kinds of science, especially in the field of chemistry and biology. But at the point right now, when we're thinking about protein design and protein self-assembly, we still have no really straightforward or simple solution for generating these kinds of structures in a completely de novo way. Even the best iterations that we have require still some kind of human to come in and say, look, these motifs and these motifs seem like they're doing something, let's try them again in a kind of semi-empirical format. And so one can imagine that if we could find new ways of interrogating protein structures, or ways to kind of add accurate, reliable omics-driven data or other forms of structural data to help to feed these kinds of advanced computational models, then in the near future, we might be able to actually better predict the impact of how even small changes to protein side chains, or maybe larger changes to mutations or multiple mutations or PTMs, might actually be able to influence this kind of design aspect. And so when we think of how we are building our data sets to look at these problems, I'm showing here kind of the two classic or conventional methods for structural elucidation. The point is you want to kind of get somewhat of an atomistic model. It doesn't have to be perfect per se, but it has to be able to tell you a little bit about what the positions are of each atom in that structure. And so on the very left here, you're seeing X-ray crystallography, which many people I'm sure are very familiar with, starting with a crystal, get a diffraction map, try to fit as much as you can and refine. And the right, you'll see NMR, and this is, although a less utilized method for biomolecules, it's still one of the gold standards. The idea is that you can look at structure, you can look at kinetics, you can look at folding, and what's highlighted in these two red boxes over here are kind of the major limitations in both approach. It's not necessarily that either approach themselves in a vacuum are really that poor of a method. It's the reality is that there's kind of a realistic trade-off between in-depth structural detail and throughput. It's being able to look at X-rays for solid state, or even cryo-EM structures for solid state, and refine and refine and refine and keep looking at multiple different models all the time is extraordinarily time-consuming and laborious. And you can only really do this in the hands of very few experts or labs or resources that are set up for this. NMR also suffers from the same kind of issues, where it's in some sense could be even slower than what you'd imagine you can achieve through cryo-EM or X-ray. But at the end of the day, you still need to assign every single peak here. And so there's kind of this kind of mismatch at the moment between how much more detailed do we need to be and how much that we can really say using classical methods. So in the current state of the art, if somebody were to try to design a new kind of protein assembly, or even try to figure out how protein assemblies come together, they're going to be met with this kind of misfit. Do I go with the X-ray and do this one crystal at a time? Do I do this with NMR and spend days to weeks to months maybe solving a single structure? Maybe this could even be somebody's PhD. And so what I wanted to do when looking at a problem like this is think of clever ways that we can now maybe introduce new or different biophysical characterization methods. And I thought that native top-down or native mass spec in general is kind of a very ideal candidate to not necessarily replace those techniques I had mentioned before, but to be a direct complement. And so for those of you who might be familiar with the concept, so native mass spectrometry, first shown by Joseph Liu in the 80s and 90s, and then first coined by Albert Heck later on in the early 2000s, is a technique where you try to spray a protein in as close to a physiological context as you can get, preserving as much of the natural, native, quote unquote, functions, effectors, and interactions that are possible. And we've made a lot of headwinds in this space over the past couple decades, with many players and giants in the field showing new methodologies and new kind of techniques to be able to do this really well. And so we've gotten really good at being able to map ligand binding, protein assemblies, and understand micro and macro heterogeneity, as well as proteoforms. Even further, some labs have taken this to the LCMS timescale. So Ying's lab actually is a great example of this with a recent paper by, I think, Matt Fisher, that shows that you could even do native separations online and characterize these assemblies in real time. And so these kinds of technologies maybe present a potentially higher throughput, but still molecularly resolved way of looking at this problem. And to even drive the point further, to give you an idea of how a intact spectra might look like when you're classically looking at it from a denatured sense, this is kind of the sort of thing you'll get. If you look at intact mass spec of a specific protein in a denatured context, you'll see the classic charge state envelope mobility. If you zoom in and try to look at a very specific charge state, well, you can try to map PTMs. You can also try to understand expression. These are just kind of basic principles that we tend to do as well in bottom-up approaches at a peptide-centric level. But what native allows you to do is preserve the whole protein complex. And so in this case, instead of just looking at a little arm of the protein, what if in reality that arm actually made itself into a larger heterotrimer? We would never be able to rationalize this unless we could preserve those features. And so native approaches tend to give you this ability to now look at protein assemblies in a nuanced way and even understand how ligand binding can occur, solvent accessibility, and even further deeper measures of how proteins behave with each other in a way that kind of preserves most of the physiological function. And since Joseph Liu demonstrated this a while back, Rachel and Joe in their Nature Chemistry paper, you can even understand how interfacial interactions between two things can be teased apart using just mass spectrometry. So you could take a protein complex, break it down in different ways, but also try to figure out how different residues are in contact or not in contact. And so you can see that this could be a potentially very powerful approach when applied to these kinds of problems. And so as maybe Neil would like to say all the time, when we're thinking about exploring higher levels of protein composition, we're starting from, in some sense, the bottom-up idea of digesting proteins, and we're moving closer and closer into proteoform level analysis. But then there's maybe one more step up there, which we could think of as native proteomics. And so the hope is at some point to be able to integrate all levels of information that we could obtain from both proteins and proteoforms, as well as their complexes, and in the future. And so this brings me back to kind of the problem that I'm going to be talking about with you all today, which is the idea of rationally designing what we're calling a foldon-derived heterotrimer, all of this guided by native mass spectrometry. And I first want to acknowledge the key players in all of this. One is Dr. Jinyu Jenny Liu, who's currently a postdoc at Stanford, Professor Ying Ge, and Professor Samuel Gelman, who's currently chair now of the chemistry department at UW-Madison. And so let's start with the framework of looking at how we're going to design a heterotrimeric scaffold. And the point here is to actually make it minimal and different from anything else to even just give more opportunities. And so what we're doing here is starting with this particular structure. This is called bacteriophage T4 fibridin. So unusually, there is this bacteriophage that comes together with these beta hairpin type beta sheets. And normally when you come together in beta sheets, you're going to tend to make amyloid plaques where you bridge a bunch of amyloids, beta sheets together, and they kind of just aggregate. But this is an example of a rare case where it doesn't aggregate. It forms a very solid, very specific homotrimeric formation. And this little piece here that you're seeing, this minimal model composed of 3 protomers AAA, is what we're calling here a foldon. And the way we're going to represent this is kind of like in this triangular representation, where each circle represents the individual protomer. And these letterings here tell you whether or not they're from the same monomer or a different monomer. This is the sequence here between all of the 3 changes. And so this kind of presents itself already as a very small, minimalistic model for a homo-oligomer. And the concept we had was basically, could we take this and through some rational design process, be able to turn this into a heterotrimeric motif? And so I mentioned to you that this is already a stable homotrimer. So how exactly are we going to plan to do that? I think one concept that's kind of underlying a lot of these things when we think about the thermodynamics of protein assembly is that, yes, in reality, it's a stable homotrimer, but also there's still some kind of dynamic equilibrium between the various pieces. Just because it comes together in a stable way doesn't necessarily mean that it's always, at every moment in time, stuck together like a glue. It's still exchanging. It's just that the kinetics of that dissociation are strongly in favor of one way. But you can imagine if we can think of clever ways to manipulate this kind of dissociation, perhaps we can weaken the structure a little bit. And instead of just having an AAA kind of motif, maybe make a very different AB or ABC introduction through some clever changes. And so what I wanted to also mention here is why native mass spec becomes a really powerful way to do this. So if you look just on the left, at the top left, you'll see here is the kind of native mass spectra of a foldon if you were to just spray this natively in the mass spec. And at the very bottom is the corresponding deconvoluted spectra. And so what you see here at the top are the various charge state profiles you obtain just from spraying this foldon structure natively. In a deconvoluted sense, you'll see that it's what looks like a trimer, dimer, and a little bit of monomer. And in reality, when you quantify this, you'll see that it's pretty much 90% almost always trimeric at all times. And I feel like something like this, just being able to pick up a protein, spray it in the mass spec, and immediately quantify all of the various oligomeric stoichiometries that you could obtain with something. I think it's a really powerful way to quantify these protein oligomeric states, but also it's a really high throughput way if you consider what are the alternatives. And so with this in mind, maybe we can find some clever way to transform this stable homotrimer into something else. And so that's kind of the design problem here. How would you take this structure that's so strongly in favor of being a homotrimer and try to transform it to do something? Well, actually, it turns out in our thinking process, one of the first ways anybody would want to try this to generate two things coming together in a mixed way is to imagine, what if you take the naive approach and you just simply mixed 2 homotrimers? So you can imagine if you had protein complex A, protein complex B, both unbiased, stable homotrimers. They still exist in their dynamic equilibrium. Theoretically, if you mix them together and you try to reach a thermodynamic balance, should you see all homo or could you find something else? And so it turns out when you do this, for the most part, in a unbiased, non-interacting way, you're almost always going to arrive at the uniform distribution model. So if you take 2 very stable things and you force them to mix with each other at all times, in this particular case where we're looking at a trimer, you'll end up with a 1:3 to 3:1 ratio, mass degeneracy included. The idea is that you take A, take all B, you kind of shuffle them together. What you're going to get is exactly this distribution if there is no specific bias or preference for each of these protomers to mix itself together. So if we know that when mixing 2 things together in an unbiased fashion, we're going to get a mixture, well, this is kind of not necessarily all loss, but it's almost like a step in the right direction. Yes, we still see homotrimer A, yes, we still see homotrimer B, but looking in the middle, we actually get heterotrimeric AB, in this case AAB or ABB. We just don't necessarily have a uniquely enriched population of one or the other. And so if you can imagine that you could isolate each one of these at any moment, what will happen is it'll just split back into its normal thermodynamic equilibrium. But promisingly, it can form this to begin with. So is there a way that we can maybe think about the design scaffolding to force it to stay as preferentially one or the other? And so how we go about doing this. So there was an inspiration here that we took a little bit when you think about like introducing kind of how you make A and B come together. There's a very classic example of this that was first originally pitched in 1952 by Crick, and over time eventually leveraged by Paul at Genentech, Paul Carton, where they came up with the first kind of bispecific antibody through this knobs into holes kind of approach. And the idea is you make vulnerable mutations in a way that brings together one protein to another non-covalently, but make it so that it's so thermodynamically stable that this kind of formation is almost guaranteed. And so thinking about it from the concept of that fold-on scaffold, if you want to bring A and B together, but bring it together in a hetero way, well, could we imagine kind of from this fold-on scaffold a way we can do this? So thinking about designing a heterotrimer formation in a bias sense by introducing some kind of knobs into hole concept. So if you looked at the structure of that fold-on, it looks like this kind of beta hairpin type propeller structure. Well, in the center, there are residues that come directly in contact with each other, but they're not necessarily in contact in a extraordinarily tight way. Maybe that gives us opportunity to introduce some kind of loosening up there, and then potentially introduce a puzzle piece that fits directly in and forms a new kind of heterotrimeric structure. So that's kind of the concept of how we wanted to move forward with this. So to start, we aren't necessarily doing this blindly. There's actually a lot of studies that go into kind of understanding how side chain pairings in biomolecules can introduce potentially stable formations. And so if you look at immunogenetics, the kind of database for this, you can actually visualize a lot of the various side chains and what their contributions are, both in terms of hydrodynamic volume and in terms of their hydropathy, their tendency to be hydrophobic or hydrophilic. And so if we think of the fold-on scaffolding at this position 14 that I'm showing here at the very center, there's kind of these classes of amino acids that could be substituted that give rise to either very large apparent volume classes like phenylalanine, or even very small like alanine or glycine. And in between all of this are the various hydrophobic moieties you can introduce. And so we postulate that perhaps some kind of appropriately selected hydrophobic amino acid with a complementarity of volume classes may be able to promote this kind of biased packing pattern. So from a design point of view, the first thing we want to do is, okay, take what nature has given to us, but now as a chemist, come in with a toolkit to try to modify it. And we're starting actually with taking this fold-on structure and kind of making it a little bit more appropriate for design and for synthesis. So we're going to make this and we're going to do this. We could do this in a peptide synthesizer. We could do this in a small scale because these things themselves are not that difficult to make. These are 25-mer protomers. So you can imagine the first thing we want to do is take some of the non-essential residues. If you just flip this fold on its side 90 degrees, we modified this N-terminal right here to change it from a glycine to a lysine to enhance the solubility. And visualizing the effects of that by native mass spec, we see that it tends to mostly preserve the oligomeric stoichiometry to trimer. And so as expected, we improved the solubility, but we didn't necessarily affect any of the trimer stability. That's great. So now we have a starting scaffold that's really soluble, very easy synthetically to make in kind of in a synthesizer solid state kind of fashion. And we can do this very reproducibly. How now are we going to try to make the necessary changes to transform this structure? Well, one thing we thought that could potentially do this is actually introducing a mutation we're calling Q11E here to partially destabilize the homotrimer. So what do we mean by that? Remember, if you mixed A and B together that are really stable, they have a tendency to just stay themselves stable and not necessarily come together. But what if you can like weaken a little bit of that protein assembly? And so what I'm showing here on the left is a variable temperature circular dichromosome analysis. This is essentially the polarization of light as a function of wavelength on the left, and the polarization of light as a function of temperature on the right. So what I want to impress on you is that if you could imagine taking the native foldon structure first, introducing the G1K mutation that we just discussed, or introducing the G1K mutation plus this additional Q11E mutation, what do you see? Well, first, you see that the structure themselves, from a secondary structure point of view, tends to be mostly preserved. But when you look at the thermal stability between the foldon, the G1K, and the G1K-Q11E double mutant, you notice a massive shift, or at least a substantial enough shift in the thermal stability, where both the foldon and G1K structures, as we predicted, and as the native MS also showed, doesn't seem to change its thermal stability. It's still very stable in its own self-type structure. But that one mutation here, Q11 to E, adds a charge to that side chain. And now suddenly this repulsion effect is making it a little bit more destabilized. When you take this double mutant and you shoot it on the native and the mass spec in a native state, you'll actually see that the mass spec gives you a very similar readout to what you expect from the circular dichromosome analysis. Again, taking the double mutant, monitoring trimer, dimer, monomer, we can quantify the oligomeric stoichiometry and see that as opposed to like the 90% trimeric abundance we've been achieving, we're now reduced this a little bit. So we've dampened some of the homotrimeric stability, and now it's starting to fall apart a little bit, but not necessarily enough to kind of completely dissociate, but just enough that maybe now we can take this scaffold and start to introduce vulnerable mutations, not just at these non-essential residues, but maybe at the pivotal residue at the center that we discussed. And so one really cool thing I want to show here is how native MS can facilitate screening of these various, what I'm calling foldon triple variants. And so if you take, for example, the valine and modify that to a glycine at the central pocket that holds that foldon structure together, you immediately see an extreme impact on the homotrimeric formation. So again, monomer, dimer, trimer by native mass spec, you're seeing almost now near 90% monomeric abundance and almost no trimeric form. So we've completely ablated to some extent the trimeric formation just through the introduction of this one point mutant. We can do this again, change the valine at position 14 in the central pocket to a lysine and reanalyze and notice that, okay, it's not completely disturbed, but it's definitely even weaker now of a trimer than it was when we first introduced the double mutant. V14F, even worse, more similar to the glycine. Now it's much more monomeric than it is trimeric, and so on with V14 alanine, and so on with V14 isoleucine. And so thinking about it from kind of a pragmatic sense, every single one of these experiments can be done in essentially minutes, just directly infusing a purified protein one at a time and immediately arriving at these very informative high-level oligomeric stoichiometry measurements. And so you can think about it from the point of view of a design perspective. Making these structures are actually not that bad. A sufficiently set up lab or resource could be able to pump out a bunch of these various mutants all in a single day or in a couple days. And from the point of view of someone wanting to analyze this, say now by X-ray crystallography or NMR, you know, good luck. It's going to take a very long time to go into this level of depth and screen this variety of various mutants to even arrive at this conclusion. And you still have to battle with all of the other challenges that are associated with those techniques. Meanwhile, native mass spec enables an immediate kind of rapid structural determination between all of these various mutants. And this allowed us to be very clever. So what we're showing here is just the homotrimer version of these triple mutants. So if you take all the foldon variants and just look at them on their own in isolation, this is kind of what you'll get. But this is the landscape we want to be in. We don't want a completely stable homotrimer. We want a partially stable homotrimer where once we mix them, we can get some action. And so going back now to that concept framework where we thought of trying to modify this key residue in the central pocket with these various amino acids of specific volume and specific hydropathy, we're seeing this kind of relationship kind of appear, where as a function of volume, hydropathy, we get a relative trimeric abundance between the various forms. So it's almost like we're tuning a bit how much trimer are we getting for what mutation we've made. And so now we can access this diverse range of various trimer abundances from glycine, alanine, valine, isoleucine, leucine, and phenylalanine. And it kind of predicts in a very nice, tractable way the results that we're seeing by native mass spec are very agreeable with kind of the theoretical framework for how we're imagining this might work. And so from here, you might want to ask the question, okay, what happens now when we start mixing them? And so I'm calling this mixing and matching of the foldon triple variants. So recall, when you take homotrimer A and homotrimer B, you mix them together. This time I've kind of elaborated all of the various structural diversity you might imagine you get. It's still a 1 to 3 to 3 to 1 structure globally, but each one can be replaced in a different way. So there's combinatorial ways of doing this. But with mass degeneracy, it's about 1 to 3 to 3 to 1. All homotrimer A, AAB, ABB, and all homotrimer B. What happens when you take two homotrimers, like in the case we were just talking about, the foldon and the point mutant G1K, which we showed had no impact on the overall trimeric formation? What I'm showing you here is raw mass spectrometry data, native MS, honing in on the charge state 5 charge ensemble. So what we're going to show here first is the annotation. This is the homotrimer AAA. Here's AAB, here's ABB, and here's BBB. This is what happens when you just dissolve all of these things in solution at an appropriate 1-to-1 molecular stoichiometry compared to the both structure types. And at the bottom here in yellow, what you're seeing is what I'm labeling as a thermocycled version of what's at the top. So you can argue if you take two solutions and you mix them together, does that necessarily mean that you're going to form the thermodynamically favored interaction? You might have a kinetically trapped variant. Or if you just take two dry powders of protein A and protein B and dissolve them together, you still have to run into this issue of what are the kinetics that are resulting in that overall mixing, and how do I know that I'm actually achieving thermoequilibrium? So you can kind of get around this problem by mixing your structures, analyzing them as they were, and then introducing a thermal cycle where you kind of heat them to denature all of the interactions and then cool them to renature these structures, and then see kind of what would otherwise be the true endpoint. And in doing so, you actually see this. So this is the mixture of foldon and G1K mutant, the single mutant, at a 1-to-1 ratio. So these black markers are the control, the non-thermocycled. The yellow are the thermocycled. And what I've done here in these dash boxes is represent kind of the uniform distribution model. So this is the amount or the abundance of a specific oligomeric state if you assume that there was absolutely no bias between how these things are formed. And I hope you can see and impress on you that in this particular case, it seems very much so that the prediction from theoretical analysis aligns nearly perfectly with the experimental data. When you have two things that don't necessarily self-interact but are very stable on their own, they make this 1 to 3 to 3-1 ratio, which completely aligns with our predictions, and native mass spec is actually able to immediately access this information. Okay, so that's what happens when you take two very stable things. But as we just demonstrated, we have a kind of library, mini library, you could say, of candidates that can be very destabilized. And so how are we going to look at each of these? Well, it's actually not that bad, but I wanted to focus in on two specific examples. And so the first one, what we're doing here, the idea is that we're pairing these foldon triple variants in search of a stable heterotrimer. So we're mining now. What happens when you take the G mutant at the center and mix it with the leucine mutant at the center? Okay, so what you're noticing here, this is raw mass spec data, just like before, focusing in on charge state 5. This is just the most abundant charge state for this ensemble. Here's the 1-to-1 mixture, here's the 1-to-2 mixture, and here's the 2-to-1 mixture. What's the reason for these mixing ratios? Well, you can imagine if it's a trimer of 3 states, then it does really matter what the mixing ratio. If you want to make a heterotrimer, they're not going to come together in 1-1. They're going to come together in some bias. And so you want to assay for each of these potential biases. So if you take that glycine mutant and take that leucine mutant and then look at it, well, what do you notice? It turns out it forms predominantly homotrimer. And this is regardless of the specific bias and how much of one or the other protomer you introduce. So if you take the 1-to-2 example, where you have double the amount of the L mutant and 1 parts of the G mutant, you still see that it's pretty much mostly homotrimer BBB. And in the same case, you reverse it. There's a little bit of heterotrimer now, but you're still mostly homotrimer to some extent. If you add up all of the homotrimer contributions, it's almost 50-50. And so you don't really have a unique bias. So there's nothing here that points to something that's completely set in one way. But looking at this glycine here, which has basically nothing at the side chain, what happens when you do one more little addition? You take a very small, super minimal addition, you replace the G with an alanine. Now what happens? Well, everything changes. So you take the same 1-to-1, 1-to-2, and 2-to-1 populations that you saw above, but now you look at the mass spec data, and what you're visualizing here is, okay, almost no homotrimers, but it's AAB. Okay, that's interesting. What happens when you do it in the reverse stoichiometry? Okay, you get a biased representation. And what happens when you do it in the ideal case, the 2-to-1 stoichiometry, you get essentially all AAB. And so the concept here is that it's an extremely small change, just one more addition of a carbon here, but it's enough potentially to disrupt the homotrimeric packing that the V14L would otherwise produce. As we saw in the above case, the V14L triple mutant almost always wants to be a homotrimer, and the glycine is just not enough to do any damage to it. But just introducing that one carbon from that alanine is all it takes to completely disrupt V14L packing and now introduce this very stable, potentially candidate heterotrimer. And so what could we do from here? Looking back now at this structure, we can take it and do a little bit more of a deeper analysis. And so what I'm calling this is the identification, but also the characterization of this new heteroligomer type structure. So if you mix this in, again, this 2-to-1 ratio, at the top, as you're familiar, you get the distribution of trimers, dimers, and monomers, and then you get the thermocycle distribution of trimers, dimers, and monomers, just as before. When you take a zoom in and you try to look at the trimeric formation, what are the players that are involved? What I've done here now is kind of try to illustrate from the conceptual framework what kind of structure packing might be occurring here. So in the homotrimer case with the alanine, you just have this one carbon at the very center. Okay, that's fine. And in the other extreme case, if you look at the lysine case, you have the leucine case, you have kind of 3 BBBs kind of packing together. Well, that can happen. It's hydrophobic interaction. And in the ABB case, it's kind of a little bit of a 2-to-1, but it's a 2-to-1 and it's sort of a mismatch way. But in the AAB case, at the top right over here, you're almost seeing what appears to be that exact kind of hypothetical knobs into holes kind of structure that we were talking about, where these 2 alanines kind of fit very neatly into a central hydrophobic core that's matched really well with that leucine. And so it turns out, if you look even further at the thermocycle data, that delta M that we're seeing here, this mass difference, is exactly the mass difference between an alanine and a leucine. And so if you were to now try to quantify the oligomeric stoichiometry, you'll see that it's still, for the most part, very highly abundant trimeric species, but the heterotrimer stoichiometry now, doing the same exercise as we did with the foldon G1K mutant, instead of being a kind of uniform 1-3-3-1, the way I've drawn these dashed lines is to account for the fact that we're not mixing them in a 1-to-1 mixture, we're doing a 2-to-1 mixture. But trust me, the math here is as it should be. And the idea is that, okay, does it follow what we'd expect from an unbiased distribution of a 2-to-1 mixture? And it turns out no. It's kind of screaming this very, very large heterotrimeric formation right here. And so that's extremely exciting because this would be the first representation of a minimal motif that satisfies exact heterotrimer formation. And so to go even further, we wanted to complement our native mass spec results by looking at whether or not we can produce a stable structure and look at the atomic level resolution of each of these interactions. And so we did just that. So when you were to structurally elucidate this, not only does this heterotrimer form spontaneously and we can visualize this by native mass spec, but when you try to produce crystals with this, it turns out that all the crystal structure types are dominantly just AAB heterotrimer. And so what I can show you here, if you look, this is kind of a schematic representation of the crystal structure we obtained from this. Here's A, and here's B, the very different residue, and I've color-coded it here. This is the alanine, leucine, right at the very central hydrophobic core. And so this already confirms to us that a lot of what we were seeing by native mass spec at least has a lot of precedence for informing how we do this design. But on top of that, I think this is one of the coolest experiments I've ever done, at least from a conceptual way. There's a lot of cool experiments out there. But what happens when you take the single crystal that made that heterotrimeric A-B structure, and you did something really simple. You were just, okay, I have the crystal, I analyzed the crystal, I think it's all good. And I was talking about this with Jenny, Dr. Jinyu Liu, and I said, well, what if we take that crystal and we just tried to see what it looks like on native MS? And so we did. So here's the inset here showing a picture of the single crystal that we took. This is one single crystal. You can imagine the concentration of this is extraordinarily low, but you just dissolve it in native-like buffer, ammonium acetate like usual, and you shoot it on the mass spec, and what do you get? You actually get exactly this distribution. It's a little bit salty, that's fine. You need that to grow a good crystal, but you see here that it's still the dominant AAB. So this is kind of native MS of the heterotrimer single crystal in a direct fashion. And on top of that, we know that this is what it is, but we can also go a step further and apply some complex up style moieties to figure out, okay, what is the structural composition of this? And so if you were to now isolate that AAB form, you get this intact AAB representation, beautifully isotopically resolved. But then you're able to dissociate them into monomeric compositions, so protomer A, protomer B, and we're now able to quantify, well, what did that really look like? It turns out you get exactly a 2-to-1 ratio directly from native mass spec. So not only does the X-ray structure give you this 2-to-1 dominant heterotrimer, but you take the same crystal, you dissolve it, you then put it in the mass spec, you get back the exact same stoichiometry of 2-to-1. So I thought this was a very beautiful representation of how closer and closer we're getting to transforming a mass spec, which we normally don't think about is capable of all of these structural methods, to something that can really be a biophysical tool and something that can kind of expand the toolkit of a lot of scientists outside of the field of analytical chemistry to find useful ways of implementing this. Beyond just for cryo, we can even think about how this could be used for guiding the rational design of new structures. And so I wanted to give a summary of the major findings. So as I mentioned, prior efforts to design heterotrimers with specific stoichiometries have tended to focus on just alpha helical, coil, and collagen-like helices. Well, that's just because that's all we kind of know from nature. And when we use even advanced computational models to figure out new cases, we actually are very limited in that we don't know what we don't know, and we can't train data sets on data that we don't have. And so new opportunities like what we're presenting, this foldon derived system, can potentially provide a very fruitful alternative to support new geometries and structure types among self-assembled proteins, and even try to understand and rationalize how even the smallest of changes can give rise to really large structural differences at the macro level. On top of that, I think this is a really wonderful way that shows the power of native mass spectrometry for rapid and quantitative analysis of protein assemblies in a way that we tend not to think about. So instead of using this as the endpoint, we're using this as the screener. We're using this at the front end. And so I hope that future efforts can maybe even leverage scaffolding like this. You can imagine now to append unlike proteins together. So you have an AAB, well, you can now bring together two things that normally don't even talk to each other. But because you have this underlying scaffolding, you can start to study how those things might interact, how you could maybe develop new glues, and how you could facilitate new designer constructs for new applications. And so with that, I'd like to acknowledge, first of all, my postdoc mentor, Dr. Carolyn Bertozzi, Dr. Jinyu Liu, who's the co-first author on this work in the Cegelski lab currently at Stanford, the Gelman lab, Sam Gelman and his lab, and of course, Ying's lab, the Ge lab. And I really want to thank my funding source, the Damon Runyon Cancer Research Foundation, that provides me the support to be able to do all of this work. And of course, I want to acknowledge the rest of the Bertozzi group here at Stanford. It's a very large, diverse set of people, and it's been wonderful working with them. And I hope this story presents kind of a very different, but also, I think, really cool way of how mass spec can infiltrate these kind of chemical biology spaces and find new use and kind of new purpose in these kind of structural design methods. And so with that, thank you so much for listening, and I'm happy to take any questions.
Great job, David. Very fascinating work. And I should clarify, this is actually David's idea. And he is very creative, talented scientist, as you know. So now we open time for questions. Anyone who has questions, you can just unmute yourself or, turn your camera and ask.
Maybe I can even stop sharing.
Okay, so Joe has a question. Nice work, nephew David. Could AlphaFold be used to confirm the mutations that leads to stable heterotrimer?
Oh, sorry, I was playing around my Zoom. Okay, yes. So it turns out, Uncle Joe, no, in fact, you cannot use AlphaFold to predict this. Actually, this was the very first thing we attempted. In fact, when David Baker had published his obligate ABC heterotrimer paper last year in Nature, there's actually a big conceit here. It's that when you wanted to make a new structure and you fed these like kind of motifs into AlphaFold, they just regurgitate the exact same motif, but maybe like slightly different in the way that they're coming together. So if you took exactly those structures I showed in all of those screens with all the various point mutants, what you'll find is they all look exactly like foldon, and they'll never change because it's so small that one perturbative mutant, all of AlphaFold, even AlphaFold 3 currently, will just say, oh, it's going to be the same. It'll never be heterotrimer. It's all homotrimer. It just looks exactly like what already exists. And so this brings up a kind of deeper point, which is we have these really powerful computational methods, but we just don't have really accurate and data sets that show what the impact of very small changes can make. And so hopefully in the future, as we do more and more of this, and as mass spec enables these avenues, maybe we'll be able to build, new data sets that show people what do PTMs do to protein structure, what do small mutations do to protein structure, and then maybe the next generation of AlphaFold will be able to start to predict this better.
Great. And I also want to make clarification that Uncle Joe and nephew David are not biologically related. They are related through the McLafferty family. Okay. So we have next question. It's great presentation from Martin Kadin. How far are you from making an ABC heterotrimer? What's the trick?
Oh, this is a wonderful question. So it turns out we can make an ABC heterotrimer, and the trick is actually kind of similar to this. So you still can screen as we just did between heterotrimeric formations, but you start with the 2-to-1 scaffold and then try to introduce one more mutant on top of it. In the current story, we didn't really get into the details of that because it was already kind of interesting enough that we have this rational design motif. But you can apply a similar kind of trick to do this in ABC way. In fact, you could argue a really kind of a naive ABC is to just modify one of the residues that are non-essential to the binding. So remember that G1K mutant at the very start that was there to enhance solubility? It doesn't make it in the central pocket. So in some sense, you could completely change or elaborate one of those residues, and suddenly it's an ABC heterotrimer already. And so that's kind of one of the underlying tricks. Now that we know that this is the core residue that matters, if you don't perturb it and you just change everything else, you can actually start bringing together ABC structures, because nothing tends to matter at the very minimalist scaffold, but you could put together ABC proteins together at the non-essential ends.
Great. I see Jake also has a question. Jake, do you want to ask?
Yeah,
Hey, Jake.
hi David. How's it going, buddy? Great talk.
Good. Thank you.
So I have a very quick question, then a longer question. What what instrument did you use for that work?
Oh, so this originally started on one of my favorite instruments, which is the FTICR.
Beautiful.
Great, great.
Yeah.
Love to hear
But
that.
you don't need an FTICR to do this. You can absolutely do this on a TOF or anything you want, you know. So, you know, it's
Yeah.
kind of an overkill, but it's a beautiful instrument.
It certainly is. And that actually leads to my other half of the question, where you had that hybrid approach where you'd use the FTICR for like deep kind of characterization, then use ion mobility or timsTOF for some structural information. So for this
Right.
type of work, what do you think ion mobility could buy you, as you would say? Like how would that data kind of feed into some of the structural kind of stuff that you're learning?
Oh, that's a very interesting question, Jake. So I think it remains to be seen from kind of the protein level, what kind of features we can do with ion mobility and what kind of new applications we have. It's still very virgin territory. And the extent that we've been able to see that, we've already shown how homo-oligomeric things look like in TIMS, and we've seen how model hetero-oligomeric things look in TIMS. But you can even imagine for ion mobility in general, could you use that as a way to inform the kind of subtle changes that are occurring? Because there's a lot of times where some of these mutations I showed in this presentation, it's a very subtle change. And honestly, it doesn't always give rise to an extremely like understandable mixture of trimer versus non-trimer. And I think a lot of that is because, you know, things might be changing at the global structure level, but it's still just held together well enough that you will see it just as a trimer, dimer, or monomer. With an ion mobility dimension, you might be able to get an understanding of how that conformer is changing as you're doing these kinds of rational design elaborations. And so maybe a point mutant or a post-translational modification or something minor can actually have a really large impact on the conformer in the gas phase. And we can better leverage that information using ion mobility. So I think there's a lot of ground there. It's just not something that was explored in the current case.
Sounds like good work for you.
Yeah. No, indeed. David
thanks,
did a great
David.
job. He got the idea and finished it. So this is one of his side projects. He's very productive in a PhD with over 30 publications and 6 first author papers, with one more is still in the process. So it's incredible. Any other questions from the audience? Okay, if we don't have questions and we pass 11 o'clock, we'll just conclude this Proteoform Thursday. And I also want to call the young scientists or, you know, the senior scientists out there in the audience, if you are interested in participating in the Proteoform Thursday, please contact Paul Dennis or any of us on the consortium board. Yeah. Please let us know. And I look forward to see all of you at ASMS. All right. Bye-bye.